Welcome to this lesson on the process of cell division. Today we will explore the intricate and vital steps that allow cells to divide and create new cells. As seen in this image of cells undergoing division, this process is fundamental to growth, development, and repair in all living organisms. By the end of this lesson, you will understand the key stages of cell division and how they ensure that genetic information is accurately passed on to the next generation of cells. Let us dive into the fascinating mechanisms of these essential biological processes. Let us take a look at our learning objectives for today. By the end of this presentation, you will be able to explain the role of chromosomes during cell division, describe the main events of cell cycle, detail what happens during the phases of mitosis, investigate how daughter cells split apart after mitosis. To begin, let us remember that the cell cycle is the process by which cells reproduce. Take a moment to think of as many words as you can that are associated with copying, such as duplicate, replica, or replicate. These words help us understand the essence of the cell cycle, the copying and transferring of information to produce new cells. Chromosomes are essential structures in every cell, serving as the storage units for genetic information. This genetic material or DNA contains the instructions that guide all the cell's activities and functions. In prokaryotic cells, such as bacteria, the DNA is packaged differently than in eukaryotic cells. Most prokaryotes have a single circular chromosome, unlike in eukaryotic cells, where chromosomes are housed within a nucleus. The circular chromosome in prokaryotes is located in the cytoplasm, freely floating within the cell. This simple structure allows prokaryotic cells to efficiently replicate and carry out the processes they need to survive and reproduce. As we continue, we will explore how chromosomes play a crucial role in the process of cell division. Now let us shift our focus on eukaryotic chromosomes. Unlike prokaryotes, eukaryotic cells package their DNA into multiple chromosomes. These chromosomes are highly organized structures that allow large amounts of genetic material to fit within the nucleus. At the most basic level of chromosomes are composed of a DNA double helix, the molecule that carries genetic information. The DNA is then wrapped around a histone uh, proteins, forming a structure known as a nucleosome. These nucleosomes act like beads on a string, compacting the DNA. When nucleosomes are tightly packed, they form coils, and these coils can further twist into supercoils, creating an even more compact structure. At the highest level of organization, we will see the duplicated chromosomes, which consist of two identical sister chromatids held together at a central region called centromere. This structure ensures that DNA is properly distributed during cell division. These levels of organization, from the double helix to fully duplicated chromosome, illustrate the remarkable way eukaryotic cells manage and protect their genetic material. As we continue, we will see how these structures play a crucial role during the process of cell division. Let us take a closer look at the prokaryotic cell cycle, a process known as binary fission. This diagram illustrates the steps involved. The process begins when the cell grows to a certain size. At this point, the cell begins to duplicate its DNA, as shown in the second step of the diagram. Once the DNA have been duplicated, the two DNA molecules attach to different regions of the cell membrane. The cell then begins to indent and pinch inward, as shown on the third step. Finally, the cell fully divides, forming two genetically identical daughter cells, as shown in the last step. Although this diagram is not drawn as a cycle, it represents a simple cell cycle. The process starts over as each new daughter cell grows and eventually. Let us take a look at the eukaryotic cell cycle, which is more complex than the prokaryotic cell cycle. This process ensures the proper growth, replication, and division of eukaryotic cells. Cells spend most of their life in interphase, the longest part of the cell cycle. Interphase is divided into three phases: G1, S, and G2. During the G1 phase, the cell grows and carries out normal functions. This process begins right after mitosis is completed and ends when DNA replication begins. In the S phase, DNA synthesis occurs. The cell replicates its chromosomes, ensuring that each new cell will have a complete copy of the genetic material. Finally, the G2 phase is when the cell prepares for division. It produces the molecules and organelles needed for mitosis. This is typically the shortest phase of interphase. Following interphase, the cell enters the M phase, or mitosis, where the nucleus divides. Mitosis is followed by cytokinesis, where the cytoplasm splits, forming two new cells. Students often think that cells get smaller with each successive division. This is not true because cells grow during interphase. The cell cycle maintains balance, preventing cells from growing too large while ensuring that new cells are functional. By the end of this cycle, two identical daughter cells are formed, ready to begin their own cell cycles. As we continue, we will explore the events of mitosis in greater detail. Now, let us focus on the M phase of the cell cycle. Unlike interphase, the M phase is much shorter, but it plays a critical role. This phase results in the formation of two daughter cells, ensuring the continuity of life. The M phase begins with mitosis, where the cell's nucleus divide. Mitosis itself can be broken down into four distinct phases, each contributing to the separation of the duplicated chromosomes. These phases set the stage for the next step, cytokinesis. During cytokinesis, the cytoplasm of the cell divides, resulting into two genetically identical daughter cells. These new cells then enter their own cell cycles, starting again with interphase. As we move forward, we will examine the details of mitosis, the first step of the M phase, and the remarkable coordination involved in dividing a cell's nucleus. Now let us examine what happens during prophase. The nucleus condenses and the chromosome become visible. Each chromosome have sister chromatids connected at a region called centromere. The spindle begins to form as shown here, with centrioles organizing the spindle fibers. These fibers will later play a critical role in separating the sister chromatids. The nuclear envelope begins to break down, marking the very end of prophase. This allows the chromosomes to move freely within the cell and interact with spindle fibers. Each structure in this diagram is essential for ensuring that the cell can divide its genetic material accurately. As we move to the next phase of mitosis, we will see how these components work together to complete cell division. Let us move to the second phase of mitosis, metaphase. During this phase, the centromeres of each duplicated chromosome line up across the center of the cell, forming what is often referred to as the metaphase plate. This alignment ensures that each daughter cell will receive an identical set of chromosomes. Additionally, spindle fibers extend from the centrioles, connecting each chromosome to the two poles of the spindle. These fibers will play a key role in separating the chromatids during the next phase. Metaphase is critical for ensuring the accurate division of genetic material. This precise alignment of chromosomes along the metaphase plate is key to the success of the next phase, where the sister chromatids will be separated. In the third phase of mitosis, anaphase, the sister chromatids, which were previously joined at the centromere, are separated and begin moving toward opposite poles of the cell. At this point, each chromatid is now considered an individual chromosome. The movement of these chromosomes is facilitated by the spindle fiber, which pull them toward opposite ends of the cell. This ensures that each new daughter cell will receive an identical set of chromosomes, maintaining the genetic integrity of the organism. Let us consider the importance of this step. Anaphase is a critical phase in mitosis, as it sets the stage for the final separation of the cell into two genetically identical daughter cells. Let us now move to the next stage of mitosis, where these chromosomes are fully reorganized into two new nuclei. In the final phase of mitosis, telophase, the cell begins to reorganize itself in preparation for the final step of division. During this phase, nuclear envelopes reform around each set of chromosomes, creating two nuclei within the same cell. The spindle fibers begin to break apart as they are no longer needed. The cell membrane begins to pinch together in the middle, preparing to separate the cell into two. This phase marks the completion of mitosis, but it is important to note that the cell itself has not fully divided yet. That step is completed during cytokinesis, which follows telophase and splits the cytoplasm, resulting in two identical daughter cells. Let us now move to the final step of cell division. Now we have reached cytokinesis, the final step in cell division. This step typically occurs during telophase and is crucial for completing the division of one cell into two. In animal cells, the cell membrane pinches into the center to form two new daughter cells. In animal cells, the cytoplasm is divided by drawing the cell membrane inward into two parts separate. However, this process is different in plant cells due to the presence of a rigid cell wall. In plant cells, a cell plate forms in the center of the cell. Over time, this plate develops into cell membranes, and eventually a new cell wall forms between the two daughter cells. With the completion of cytokinesis, the process of cell division ends, leaving us with two genetically identical daughter cells. This cycle ensures the growth, repair, and maintenance of multicellular organisms. Let us now review the mitosis overview diagram, which summarizes the process we have just explored. While this diagram includes interphase and cytokinesis, remember that these are not technically part of mitosis. Mitosis specifically refers to the division of the nucleus. Now let us identify and describe the four stages of mitosis, starting with stage 1, prophase. The chromatin condenses into visible chromosomes. The centrioles separate and the spindle begins to form. At the same time, the nuclear envelope breaks down, allowing the chromosomes to move freely. Stage 2 is metaphase. The chromosomes align along the center of the cell, called the metaphase plate, with spindle fibers connecting the centromeres to the poles. Third, anaphase. The sister chromatids are separated and pulled along the spindle fiber toward opposite poles of the cell. These chromatids are now considered individual chromosomes. Fourth, we have the telophase. The chromosomes arrive at opposite ends of the cells and begin to decondense, losing their distinct shapes. Nuclear envelope reform around each set of chromosomes, completing mitosis. Finally, let us discuss what happens during cytokinesis, the last step in the cell division. In animal cells, the cytoplasm is divided by pinching the cell membrane inward, forming two identical daughter cells. In plant cells, a cell plate forms and develops into a new cell wall, separating the two daughter cells. With this review, we can see how the process of mitosis ensures the accurate and organized division of genetic material, maintaining the continuity of life. To conclude this lesson, we have explored the intricate and organized process of mitosis, a critical mechanism that ensures the accurate division of genetic material into two identical daughter cells, from the preparation in interphase to the final separation in cytokinesis. Each phase plays a vital role in maintaining the integrity and functionality of living organisms. Mitosis not only supports growth and repair but also upholds the continuity of life by replicating cells with precision. Understanding mitosis helps us appreciate the complexity of cellular processes and their importance in both normal development and their prevention of abnormalities. As we move forward, let us continue to think about how these cellular events connect to larger biological systems and the role they play in sustaining life. Thank you for your attention and engagement in today's lesson.